Hello friends, hope all are fine. Welcome to my channel Sam Biology Class. Today we will discuss about the principle and applications of scanning electron microscope. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. A scanning electron microscope or SEM is an advanced imaging tool that scans a beam of electrons across the specimen surface and generates a highly magnified three-dimensional image. The resolution of SEM is about from 1 to 10 nanometer. It can magnify the specimen from 1 to 2 million times. The first SEM was developed by a German physicist Manfred von Andrén in 1937. This technology was further refined in 1942 by Vladimir Zhurkin to provide spatial contrast. Like transmission electron microscope, SEM also uses electromagnetic lenses to focus the electron beam. But unlike TEM, in which a beam of fast-moving electrons is transmitted through the sample, SEM sweeps a beam of electrons across the specimen surface, back and forth, in a systematic line-by-line pattern known as raster scanning. This figure illustrates the functioning and different components of a scanning electron microscope. The first component is an electron gun. It is a heated tungsten filament that generates electrons. So the gun is the cathode. These electrons are then accelerated by applying a massive voltage between the gun and the anode. The function of the anode is to attract, accelerate and shape the electrons into a fine beam, which is then projected by the electromagnetic condenser lenses onto the specimen. Generally, there are two condensers. The first one is a strong lens which demagnifies the electron beam. It shrinks the large beam into a tiny intense spot. The second condenser, which is a weaker one, controls the beam spread and intensity. Its function is to project the demagnified electron beam onto the specimen. It dictates whether the electron beam will be focused as broad parallel illumination or as fine tight probe. So these two condensers work together to control the beam intensity. Next to condenser lenses, there is an aperture. The aperture is a precisely drilled metal disc which physically blocks stray off-axis and scattered electrons from entering through the scanning coil. After passing through the aperture, the electron beam strikes a scanning coil. There are two coils, X coil and Y coil. The function of the X coil is to sweep the electron beam across the specimen surface horizontally from left to right. On the other hand, the Y coil sweeps the electron beam vertically down the samples, forming the raster pattern. The scanning coils are controlled by scan generator, which functions as the brain, and the coil is the muzzle. Both of them work together to sweep the electron beam across the specimen surface in a systematic line-by-line pattern. The function of the scan generator is to continuously track the x and y coordinates of the electron beam and sends this positional data to the system. The scanning coil and the generator work together to ensure that the exact position of the electron beam corresponds to the specific pixel on the viewing screen. In raster scanning, the electron beam moves in a systematic line-by-line rectangular pattern from top to the bottom. First of all, the electron beam moves from top left corner to the right. This forms the first scan line. When the beam reaches the right end, it is turned off and returns back to the left below the first line. This returning back to the left is known as horizontal retrace. Again, the beam is turned on and then moves from left to right. This forms the second scan line. When the beam reaches the right end, again it is turned off and returns back to the left below the second line. Again the beam is turned on and then moves from left to right. In this manner, scanning takes place in a systematic line-by-line pattern from top to the bottom. The electron beam moves from left to right. When the electron beam reaches the right bottom corner, it is turned off and returns back to the top left corner. This returning back is known as vertical retrace, which is shown as black line. Blue lines are scan lines and the red lines represent horizontal retrace. So in this way, the scanning electron microscope generates a highly magnified, more detailed, pixel by pixel grayscale image on the viewing screen. In scanning electron microscope, below the scanning coil, there is an electromagnetic objective lens. The objective further condenses the electron beam, which is then focused on the specimen, which has been placed on the support grid. For scanning electron microscope, specimens must be conductive. As biological specimens contain water, they are non-conductive and are prone to collapse under the vacuum. First of all, the specimen is treated with chemical fixative such as glutaraldehyde or osmium tetroxide to preserve the structural features. The specimen is then subjected to dehydration through a series of ethanol or acetone wash and is then dried gently. The dried sample is then coated with gold to ensure conductivity. After that, the specimen is placed on the support grid for examination. Other types of samples must be prepared according to the procedure subscribed. When the electron beam strikes the specimen surface, two types of signals are generated. One is secondary electrons and number two is backscattered electrons. Secondary electrons, which are of low energy, are emitted from the top surface layer of the sample. They cannot escape from deep within the specimen. The secondary electrons are then captured by a detector which converts them into a weak electrical signal. This weak signal is then amplified. The function of the scan generator is to synchronize the positional data of the electron beam that includes its x and y coordinates with the amplified electrical signal and generates a highly magnified three-dimensional image on the viewing screen. As the secondary electrons are released from the top surface layer of the sample, they are used to study the surface topography and morphology that includes texture, shape, and external structures present on the specimen. Raised areas and edges of the specimen emit more secondary electrons. Those areas which are bright in the screen. On the other hand, deeps and shadows which release less electrons appear dark in the image. The other type of signal is backscattered electrons which are of high energy. These high energy electrons are captured by another detector and are used to distinguish between different regions of the sample based on their elemental composition and atomic number. Heavier areas of the specimen which consist of elements with high atomic number appear bright in the image. On the other hand, lighter areas appear dark. Scanning electron microscope is an essential tool across scientific and industrial fields. It has several applications. Number one, it is used to study the surface topography and morphology of different types of samples. Number two, in material science and engineering, SCM is used for checking cracks, to examine microstructures, to analyze alloys, nanotubes, and semiconductor wafers. In life sciences, it is widely used for imaging cells, tissues, bacteria, and virus. In addition to this, in forensic science, SCM is used to analyze evidences such as metal fragments, paints, gunshot residues, fibers, and hairs. So friends, this is all about the principle and applications of scanning electron microscope. For more updates, subscribe my YouTube channel, Sam Bal.